Does this is working, is it? Yes, it is, isn't it? Good. Well, thank you all for coming. I understand England are playing right now. I'm not sure if they've finished, so I'm impressed to see you all here, though I did hear it was a terrible match earlier on. No surprise there. So, I have a very different perspective to Matt Powner on the origin of life. In fact, I am coming at it from a background in bioenergetics, and that's where I'd like to come at it this evening. So this is what's going on in you right now. These are your mitochondria, which are the powerhouses of the cell. This is where you're burning up food in oxygen. And this is what's going on in a nutshell. So we are stripping electrons from food and you're passing them down. This is a membrane here. This is the same membranes that you're seeing here. You have a kind of a wire within this membrane, and electrons are flowing down this wire to oxygen. So you have a continuous flux of electrons to oxygen, reducing oxygen to form water. And that current of electrons is extruding protons across the membrane. And so we have essentially a reservoir of protons on this side of the membrane. And it's the flow then of protons through a turbine in the membrane. This is the ATP synthase enzyme. And the flux of protons, almost like the flux of water through a hydroelectric dam, drives the formation of ATP, which is usually called the energy currency of life. Essentially all cells, all life on Earth is using ATP to power everything about the cell. Now what's really quite surprising about this is how universal it is. It's not just us, it's bacteria, it's basically all life on Earth is doing this. Now these, I've just shown these proteins as little blobs here just to simplify everything. Well, that's what they look like in reality. Just to give you some sense of how, so this is the ATP synthase again here. These are enormously complex proteins. I'm not going to go through what all these structures actually are. This is the membrane here, so they're all sitting within the membrane. This is what's called the respiratory chain, and this is conducting electrons down to oxygen. We're missing one here, that's the one that's missing. This is even larger, this is like a steam engine in fact, so this shuttles backwards and forwards, pumping protons across. So we have an enormous level of complexity about how respiration works. But what's really surprising, two things that are really surprising. One of them is how it works. Why on earth does all life pump protons across a membrane? Why does it do that? Why not just chemistry? It does something really rather odd. And the second thing is, it is enormously complex, but at the same time, it's universal. It's actually this form of generating energy as ATP, so forming ATP, is as universal as the genetic code itself. It's all life is doing it this way, which seems to be of fundamental importance, but it's hard to see how it could have arisen because these proteins are so complex and so sophisticated, and the membranes that they're embedded in are also very complex, very sophisticated. It's such a counterintuitive mechanism. It was discovered by Peter Mitchell, that's him in 1946 in Cambridge. He called it the chemiosmotic hypothesis, and it was he published that first in 1961, so we're going back quite a long time. When there was a millennial edition of Science, I think, and Leslie Orgel, who was one of the pioneers of origin of life research, who knew Mitchell back in the 1940s and early 1950s, wrote, "Not since Darwin has biology come up with an idea as counterintuitive as those of the great physicists of Einstein, Heisenberg, Schrödinger." It really is a strange idea. And that's my motivation in this area. Where did it arise? Why did it arise? Now, if this is a fairly standard, relatively old tree of life, we can discuss trees of life later on. The only point I want to make here really is that the deepest branches on this tree of life are between the archaea and the bacteria. The eukaryotes up here, that's basically all of us and all complex plants and animals and so on. They're almost irrelevant to what was going on down at the origin of life. And there's these deepest branches, they're all autotrophs, which is to say these are bacteria which are generating their own carbon, their own energy and so on, basically not from the sun, not in photosynthesis, but from the rocks around them. So they're using inorganic molecules to produce their own organics. So that's autotrophy. They're often thermophilic. They're living in hot springs or in hydrothermal vents and so on. And they're all chemiosmotic, not only for their energy, but also they require this chemiosmotic coupling. They require these proton gradients across membranes to drive carbon fixation as well as energy. So it's two things that it's doing for them. Now, if I just come back here a moment, I imagine a lot of people these days will have heard of the archaea, but not necessarily everybody. So the archaea look like that, and the bacteria look like that. You'd never be able to tell the difference. In fact, we've known about some archaea for 300 years or more. Methanogens, for example, are found in swamps and so on, and very often the swamp gases will spontaneously ignite in the presence of phosphine or something. So we've known about methanogens for a long time. We didn't know that they're archaea, and you can see here that in terms of their genetics, they're really rather different to the bacteria. This is the deepest branch in the tree of life, is between the archaea and the bacteria. So in their genetics and in their biochemistry, they're very different, but in their morphology, they look basically the same. You'd never know that they were fundamentally different groups. So we can try and compare the properties that these groups have to try to reconstruct what the last common ancestor of all life on Earth might have looked like, and this is fairly standard stuff. So we can look at the traits that these different groups have, see what they share, see what they don't share. So I mean, something like photosynthesis, for example, we only find it in cyanobacteria, and so we know it arose in the bacteria. You don't find it in the archaea at all. So we do that more systematically. They share the universal genetic code. That's right across all of life. Ribosomes, so these are the protein-building factors, and I think Matt might say a little bit more about these later on. They're found right across all of life, apart from viruses, of course. Transcription, that's the way in which DNA is read off to form RNA, which is a second code. And then translation from RNA on the ribosomes to make the proteins and so on. That's basically the same. The genes involved, they're homologous. They share a common ancestor. The proteins they encode, you can superimpose them in space on top of each other. We know that they're related. There's all kinds of bits and pieces of biochemistry as well. Some of the Krebs cycle, for example, that's shared as well between the archaea and the bacteria. And chemiosmotic coupling, as I've just mentioned, and the ATP synthase enzyme. This is the turbine in the membrane that's driving the ATP synthesis based on the protons coming through it. So that's all shared. What's different, though, is really quite shocking, because the cell membrane is very different. There's almost no chemical similarity. In terms of the physics of it, it's very similar, but in terms of the chemistry, they're very, very different. And all the enzymes required to synthesize these lipids, they're different too. The cell wall is totally different in bacteria and archaea. So basically the boundary between the inside of the cell and the outside world are totally different in bacteria and archaea. Glycolysis, this is the pathway of fermentation. A lot of people thought this was perhaps the earliest energy pathway in living cells. Well, that's totally different in bacteria and archaea as well. DNA replication, you'd think what could be more fundamental than that, but it's totally different in bacteria and archaea. The enzymes involved are just not similar. And the respiratory chains that I've just shown you, they're the bacterial versions. The archaeal versions, they have some similar subunits, but most of it's different. So again, it looks as if that arose independently. So we now have this portrait of the last universal common ancestor, Luca, which is quite perplexing. Just to give you a sense of the membrane structures, this is the membranes of archaea. So they have isoprene lipids instead of fatty acids, which we have and bacteria have. They have what's known as an ether bond here, whereas they have an ester bond with an extra oxygen in there. And then this is glycerol phosphate. They both share the same head group. This is a hydrophilic head group that allows it to form a double membrane layer, but it's a different stereoisomer. And again, that raises the question, there seems to be no selective advantage to having one or the other stereoisomer there, but they've come up with it independently and differently. So again, it looks as if they do not, the last common ancestor did not have a modern membrane, whatever that might mean. So here's the paradox. Membrane bioenergetics, that's what we're doing when we're breathing oxygen and so on. They're universal, but the membranes are not universal, and neither is the active proton pumping machinery. Now, the paradox could be resolved if life actually originated in some kind of an environment where there are natural proton gradients. So you're not having to produce your own by pumping all the time. You simply use what's naturally there. Well, maybe there's a few environments like that, but the only one that I'm aware of are what are known as alkaline hydrothermal vents. Now, these were discovered, well, we've known about them for a while, but this is the most spectacular example that was just discovered in the year 2000. So this is the Lost City, which is just off the axis of the mid-Atlantic ridge. And it's not I'll show you, it's not like black smokers. Lost City is a very good name for it. It really does look as if it's been deserted. And you see these almost doodles of limestone. It's very, very ornate. And there's no smoke coming out. It does look as if it's been abandoned, but these are active vents, and there are hydrothermal fluids emanating from them. Well, they're not produced by the reaction between magma and water leading to these black smokers that everybody's familiar with. They're actually produced by a chemical reaction between olivine, which is a mineral found typically in the upper mantle, and water. So it's just a chemical reaction between rock and water, essentially. Here's olivine, and it produces serpentinite. Now, serpentinite is quite a common building stone. So what's interesting here is not the bloke in the foreground, it's the wall behind him. This is the United Nations building, and this is a serpentinite wall behind him. So that reaction really is, in my mind at least, one of the reactions that could have been driving the origins of life on Earth. And essentially what's going on is just this reaction between olivine and water to produce serpentinite, hydrogen, and heat. So it's an exothermic reaction. Basically, ferrous iron in olivine is being oxidized to ferric iron, Fe2 plus goes to Fe3 plus, by water, giving you very alkaline fluids rich in hydrogen gas. So the fact that they're alkaline fluids and rich in hydrogen is important to the overall story. If by any chance you happen to be passing home through Euston Station this evening, have a look at the floor tiles, because these are serpentinite floor tiles. So it's generally quite a common building stone. You'll find it in a lot of banks as well, but this is the biggest expanse of serpentinite that I've ever come across. So just to reinforce this difference, these are black smokers here. Everybody will be familiar, no doubt, with black smokers belching out this black smoke. They're actually iron sulfides precipitating usually. This is Lost City. This is a 1 meter marker here to give you a sense, and this is 1 meter as well. So it's larger than your average black smoker. This is Deb Kelly, who discovered Lost City in the year 2000, holding a lump of a black smoker, which you can see this fairly conventional chimney through which all these fluids are belching. And this is a lump of Lost City, which is microporous. It's a little bit like a sponge. There is no chimney. You simply have a percolation of these hydrothermal fluids through it. And you can see here, this is looking inside. So this is it's a very sponge-like rock. Now, these vents were predicted to exist on the ocean floor actually a good 10 years before they were discovered. And they were predicted by a guy called Mike Russell, who's now at NASA Jet Propulsion Laboratory. And he argued that they should be saturated in millimolar quantities. So that's quite a large amount of hydrogen. There should be temperature gradients. There should be pH gradients. So we have alkaline fluids entering these vent systems, and we have what we think would have been a relatively acidic ocean 4 billion years ago. There was a lot more CO2 then, and with a lot more CO2, it dissolves as carbonic acid. We have the problem with today with increasing CO2 levels, we get acid oceans. And also back then, when there was no oxygen, the oceans were relatively rich in ferrous iron, and so we would expect that there should be catalytic iron, nickel, sulfur minerals forming in the walls of these vents in place of the carbonate that we find today. So these ideas were a bit on the wild side until the discovery of Lost City, and Nature ran a piece which they entitled Naissance Man, as in Renaissance man, naissance in this sense meaning origins, and did a picture of him looking like Erasmus, mocked it up in Photoshop, I suppose. This is the reactor that Mike had built, and a little bit of Lost City behind him. So some of these ideas went from being really quite unknown 10, 15 years ago to being really quite relatively mainstream now, though I would not say that they were particularly mainstream. This is essentially what we have then. In a vent, we have protons on the outside. We have a thin inorganic barrier around one of these pores, and on the inside we have alkaline fluids. So we have both chemical and electrical differences across those membranes. There are some issues with this. I'll talk more about that later. And this is roughly what cells do. So here we have a very thin, substantially thinner organic membrane around the cell. We're pumping protons across it. But it's a very analogous situation. The question is, can this situation have given rise to life? There are two questions here which I'll deal with in the rest of the talk. One of them is, well, let's assume that these natural proton gradients were important. Why would a natural proton gradient do anything? There's no proteins, there's no genes, there's no lipids, no membranes, nothing here. We've just got an inorganic vent with proton gradients across natural inorganic barriers. What good does that do us? Is it any use at all? And the second question is, cutting forward, I'm going to skip all the bit that Matt will talk about later on. But if we do end up with cells in this system relying on the natural proton gradients, why on earth would they start to pump their own protons when they've got the natural ones there already? So I'll talk briefly through the first question in a little more detail on the second one. So I'm taking my insights anyway from methanogens, which are archaea, and acetogens, which are bacteria, and both of them live only from carbon dioxide and hydrogen. That's basically all they need to live. And these, of course, remember, are the gases that you find naturally in these vents. The reaction between them in the absence of oxygen is exergonic. It releases energy, and quite a lot of energy, and certainly enough for these cells to grow on. If we could do this, we could produce synthetic gasoline, and it would solve a lot of the world's energy problems, but we've not figured out a way of doing it yet. It's not only producing methane and acetate, it's actually all organic matter. So this is theoretical thermodynamics, but basically anything which is a negative value here, if delta G is negative, that means it's releasing free energy. So the total cell biomass at temperatures between 25 and 125 degrees C, it releases energy to form total cell biomass from hydrogen and CO2 alone. Nucleotides are a little bit more difficult. You have to put in energy for that. But amino acids, fatty acids, and so on, they should form spontaneously under these conditions, under alkaline hydrothermal conditions. The fact is, though, that they don't. And we can find trace amounts, really, of methane and formate and so on. But this is the problem. There's a barrier here. You have to put energy into that reaction to get up to formaldehyde here, so a partially reduced product, and then it's energetically downhill all the way. So life is somewhere around here, and so there is energy released by that reaction, but to get from CO2 to here, you've got to go over this hump. You've got to make formaldehyde somehow, and that's not an easy thing to do. Well, these cells, they use a proton gradient across the membrane, and they use this protein here, which is called the energy-converting hydrogenase. It's actually rich in iron-sulfur clusters and so on. And that reduces another protein called ferredoxin, also an iron-sulfur protein. So we've got basically a lot of iron and sulfur in here. We've got a proton gradient across the membrane, and it's reducing carbon dioxide to organics using hydrogen, driven by the proton gradient. Now these bugs have to regenerate that proton gradient, but you could cut that out with a natural proton gradient, potentially drive everything. So is there any way that we can learn anything from that? Well, possibly. So this is another way of seeing why you can't reduce CO2 with hydrogen. This is the reduction potential. So again, we can't go uphill. So this is the reduction potential of hydrogen. It simply can't do that. You have to put in energy to make that work. That's at pH 7. It will be the same at pH 8 or pH 10, any pH. But what we actually have in the vents is hydrogen at alkaline pH, pH 10, and CO2 at acid pH in the old oceans, pH 6. And under those conditions, it should actually work. Theoretically, it should work. So the question is, well, how could you possibly transfer electrons from hydrogen in alkaline conditions to CO2 in acid conditions? And the answer is, well, if you've got a very thin semiconducting barrier here, this is dying on me a little bit, I'm afraid, then it should actually work. In theory, you should be able to transfer electrons across that thin barrier onto carbon dioxide to form formaldehyde. So this is what we've been trying to do. We've built a simple desktop reactor. We're producing these little vent structures inside here, which are basically, this is McKinnowite, which is an iron-sulfur mineral. And these walls are as thin as we can make them. These are actually quite dynamic structures. And we're trying to see, we're just trying to measure, can we make formaldehyde? Well, we can make formaldehyde, but in very, very small amounts. So we're not yet persuaded that we've really made much of a breakthrough, but hopefully we can improve this system. Once we've got formaldehyde, it's really remarkably easy to go on under these same conditions to produce things like deoxyribose and ribose. But I'm not going to say more about that now, and Matt will no doubt say a lot more about that kind of chemistry. I'm going to skip through this bit. I was always amused by this cartoon where he's saying, then a miracle occurs here at step 2, and the prof says, I think you should be more explicit here in step 2. So I'm going to leave that to Matt. And I'm going to address my final question, which is, if these conditions, if these natural proton gradients can drive the beginnings of organic synthesis, then it's possible to imagine that you can go through right to protocells, right the way through to genes and proteins. So I'm not going to talk about that. But once we have some kind of cells with genes and proteins in this environment, how can they leave that environment? What's going to drive them to produce their own proton gradients? And can that say anything about this difference that I was talking about earlier on between the archaea and the bacteria, these fundamental differences? And I think the answer is it can. So this is a reminder what's happening in you. So we have a flow of electrons coming from food, going to oxygen. This constant current of electrons is driving proton extrusion across the membrane, and that's driving ATP synthesis. Trouble is, cyanide, for example, if you take a cyanide pill, it binds to complex 4 right there, and it blocks that constant flux of electrons, which is why cyanide will kill you. And so if you're not pumping out protons all the time, you've got a problem. Now, in a natural proton gradient, we should face exactly the same problem. It's a kind of a cyanide problem. We just have a natural gradient, and we have a natural turbine, whether it's this ECH that I mentioned or an ATP synthase, and so protons come through and it drives the turbine, but then once they're through and inside, how do you get them out again? So you should collapse that gradient within seconds or something, in which case it's no use to you at all. So we've built a simple mathematical model to try and look at the conditions under which these kind of proton gradients might work. And essentially what we have here is continuous acidic flow, pH 5 to 7, so this is the ocean. We have a cell which is kind of wedged here, which is perhaps a little improbable, but this is trying to idealize the situation for modeling. Exposed on one side to these acid conditions, on the other side to alkaline conditions. And we have an organic membrane around this cell. This is symbolizing a fairly leaky membrane, and I'll come to the importance of that in a moment. And these two phases are separated generally by an inorganic barrier, which is discontinuous. And so, given those starting assumptions, this is with a modern membrane, a cell with a modern membrane put inside this natural proton gradient, and you collapse the gradient completely from quite a large free energy available to 0 in about a second in this model. So it really does not work. You really cannot drive anything with a natural proton gradient and no pumps. But you can if that membrane is really leaky. So these are the modern membranes. This is the permeability of the membrane. These ones here, 10 to the minus 2 centimeters per second. I won't try to explain what that is, but essentially this is a very leaky membrane, a little bit like a fatty acid vesicle or something. And so you lose a little bit of power, but then you can sustain it. And you can sustain actually quite a large amount of free energy, which will continue to drive biochemistry forwards, so long as you've got a really leaky membrane. If you try and improve that membrane, you cut yourself off from the proton flux and you'll die. So in principle, this situation equivalent to the methanogens I mentioned, you can drive with a natural proton gradient, both carbon reduction and energy, ATP synthesis, from a natural proton gradient in a vent, so long as you have a really leaky membrane. So then the question becomes, well, how would pumping stop? So this is, here we are, this is the membrane permeability. So this is getting less and less permeable as you come across here until you arrive at modern values of membrane permeability. So we're here now, and without a pump, if you make the membrane less and less permeable, you lose power. By the time you're kind of approaching modern cells, you're losing power, you're bleeding off power. If you insert a pump into that membrane to pump protons out again, if you don't have enough, you lose power anyway. And you certainly gain nothing from having a pump. Even if you have a lot of pump, with a very leaky membrane, you gain absolutely nothing. By the time you get to modern membranes, it's better, but there is no selective pressure here. There's no driving force. You have a leaky membrane, you're doing well here. You improve your membrane a bit, you don't do any better by pumping. You improve your membrane a bit more, you have a bit more pump, there's no advantage. So we're kind of stuck here. There is no advantage to improving membranes or to originating pumps. One possible answer is not changing the composition of the membrane, but changing what we call the coupling ion. So instead of running everything off just protons, if you were to insert an antiporter in this membrane, which is just really a turnstile, so a proton comes through and a sodium ion goes out, another proton comes through, another sodium goes out. Sorry, I'm losing my microphone. So basically then you have a natural proton gradient which is capable of driving a biochemical sodium gradient. And one will add on to the other. And so initially you lose a little bit of power because protons are coming through and you're collapsing the gradient, but as each proton comes through, one sodium goes out, and over time you increase the gradient. The reason you increase the power is because sodium doesn't cross the membrane nearly as easily as protons do, and so you essentially add the two onto each other. And that does two things for you, which are really important, and this is just really coming to the end of the talk now. So one of them, because it gives you more power, on, say, a 3 pHu, so this is without any, the SPAP is a sodium proton antiporter here. So this is with a pH gradient of 3 units. This is without any sodium proton antiporter. This is with 1% of it in the membrane and 10% in the membrane. So it's giving you more power. But that also means that you could survive on a smaller gradient. So you could probably survive on a gradient of just 1.5 pH units, which is to say about 50 times less protons than you would require to survive without that antiporter. So it's allowing you to survive on smaller gradients. It's allowing you to spread through these vents, to diverge perhaps because you're no longer in direct contact with other cells. And so you can maybe take over contiguous vents and so on. You can survive in a much greater range of circumstances if you've got one of these antiporters, because it allows you to survive on a much lower natural power. And that is going to allow the beginnings of divergence between different populations. But the second thing that it does, which is key, is it means that there is a benefit to pumping. Because now, if you pump a proton out, it might come back through the membrane, but it might come back through this antiporter, in which case the sodium will go out. So for every proton you pump out, you're more likely to retain a little bit of that energy in the sodium which has gone out. And so you improve your membrane a little bit, and you get a bit of an advantage. You improve your membrane a little bit more, and you get a bit more of an advantage. And so there's now a selective pressure. As soon as you have this antiporter, there's a selective pressure to improve your membranes and to increase the number of pumps that you've got in the membrane and so on. And so from a situation where there was no advantage to pumping and no advantage to improving your membrane, we now have a situation where you have an advantage to both, caused by the same protein that allows you to spread throughout the vents. In other words, we could imagine that there might have been, from a common starting cell, the invention of this one protein allowed spreading and allowed independent origins of membranes, modern membranes, and independent origin of the membrane pumps. And that's just a reminder of the situation without the sodium proton antiporter. So just to really finish, we would picture then the last universal common ancestor as being quite an unusual cell, dependent on these proton gradients, living in alkaline hydrothermal vents with a sodium proton antiporter, with an ATP synthase, with an ECH. This is the energy converting hydrogenase, which I'm not showing here. Divergence as soon as you've got the antiporter, and then independent origins of these different types of membrane, independent origins of the different types of pump. And other differences which are very striking, for example, DNA replication, as I mentioned. Well, the DNA in bacteria during replication is attached to the cell membrane. So if the cell membrane is totally different, you can imagine that all kinds of proteins and genes would need to be different as well for DNA replication to take place attached to the membrane. So it begins to make some kind of intellectual sense, at least, of why it is that DNA replication is different. It's different because the membranes are different. The membranes are different because the only way you can survive on a natural proton gradient is by having a very leaky, modern membrane. So it makes some kind of intellectual sense. Whether that's true or not is a different question, and we have to find ways to test that kind of hypothesis. One possible way, just to leave you with this, is the distribution of these proteins. Now, this is the ECH here, and that's actually made up of several subunits in itself. And this subunit is an antiporter sitting in the membrane. It's a sodium proton antiporter. And then we have a ferredoxin-reducing subunit here, which is, again, is rich in nickel and iron and so on. This is complex 1 in your respiratory chains. So this is the first pump that I showed you at the very beginning, which is driving protons across the membrane. And it's composed of the same subunits as ECH. So this is not nearly as obscure a protein as you might think. It's just been split up here. We've had these other subunits inserted into the middle of it. These are 3 antiporters, sodium proton antiporters. So our own complex 1 in the respiratory chain is made up essentially of the same proteins that I've been talking about that I say are necessary in vents. These are found right across all of life. It's not just simply in us. It's almost as if we've got a little bit of the vent world still in our own membranes. And I think on that note, it's a good place to finish. So thank you all for listening. Thanks to various collaborators. My little group at UCL working on the origins of life is Victor Soho, who did a lot of the modeling I've just shown you, Alex Witcher and Barry Hershey, who've been working on the reactor itself. And I think questions come later, so I shall simply pass over to Matt.